The normal life cycle of a cell is divided up into three phases. Interphase, where the cell functions normally for the majority of its life. Mitosis, preparation for cell division by duplication of its genetics and nucleus. And cytokinesis, the final act of cell division where one cell splits into two daughter cells. Mitosis itself has four phases: the prophase, the metaphase, the anaphase, and the telophase. This is a plant cell in division.
The nucleolus, a densely packed section of the nucleus containing DNA, RNA, and proteins, decompresses and disappears, and the chromatin in the nucleic interior condenses into highly compacted coils known as chromosomes. So much so that the originally miles-long DNA strands are shrunk into a minuscule number of nanometers. These chromosomes duplicate themselves into identical sister chromatids. These sisters are held together by a protein known as a kinetochore, which attaches the two chromosomes at the narrow waist. Meanwhile, outside of the controlled nucleus, the mitotic spindle is forming, and its many microtubules are being assembled from the various proteins found in the cytoplasm. Suddenly, the nuclear membrane disintegrates, and the microtubules move in, attaching to the kinetochores of the chromatids, or to the other microtubules extending from the opposite side of the cell. The chromatids align at the cell's equator, directly in the middle of the two sets of microtubules. The mitotic spindle aligns itself to the poles of the cell and starts to pull the chromatids towards them via the kinetochore. In addition, the microtubules bonded to the end of the other microtubules begin to grow apart from one another while remaining attached, thus putting tension on the kinetochores and on the cell's structure itself. The tension on the chromatid's kinetochores causes the kinetochores to break, and thus the two sister chromosomes move apart to the opposite poles of the cell. The cell structure begins to stretch and lengthen due to the non-kinetochores' spindles' continual growth in an outward direction. This process continues until there is a copy of each chromosome at each pole of the cell. Once there are two corresponding chromosomes at the opposing poles of the cell, a new nucleus forms around them from the fragments remaining from the original nuclear membrane, and the chromosomes unravel back into chromatin. At this point, the process of mitosis is completed, but the cell has yet to physically divide into two cells. Now that mitosis has been completed and all of the new genetic materials are in place at the opposing ends of the cell, a cell plate begins to form at the cell's equator, the same point at which all of the chromatids were earlier aligned for separation. As the cell plate forms, the microtubules of the mitotic spindle dissolve into proteins, which disperse into the cytoplasm. Gradually, the cell plate grows until it forms a cell wall between two halves of the original cell, then the cell membranes pinch off, and the single cell becomes two genetically identical cells.